NMDA receptor function extends beyond synapses and simply the ion channel effect. So this is let me grab this. Cartoon representation that many of you may be familiar with of an NMDA receptor, the amino-terminal domain, ligand binding, transmembrane, and the C-terminal. And we're typically thinking of this in the context of mature neurons and synapses. However, NMDA receptors are also found on cells that don't make synapses. So for example, in neuroprogenitors, and our work and work of others have shown key roles for NMDA receptors in regulating neurogenesis. They're also found in non-neural cells. One of the examples I'm not going to have time to talk about today is in terms of neural crest. We found some very interesting neural crest phenotypes in our NMDA receptor knockouts, and I'd be happy to talk to people more about that after. I think it's relevant to a lot of the talks later in the session. As we've already heard about from Catherine Roche, that the NMDA receptor is more than simply an ion channel. And there are lots of this CTD or C-terminal domain that's often lopped off by a lot of the electrophysiologists, actually does some interesting things. So it's a site of interactions of many interesting proteins, like McGuck proteins like PSD95, CAM kinase 2, as well as interacts with signaling pathways like the MAP kinase pathway. So for us, the overarching questions that we've really been focusing on are, first, what are the non-synaptic roles for NMDA receptors in neural stem cells and other places? One of the other big questions that we're going to just scratch on a little bit is, or talk about some of our strategy of how we're trying to work on this, is really trying to understand how we get these various phenotypes from seemingly similar mutations in NMDA receptors. And related to that are why do gain and loss of function mutations sometimes produce the same phenotypes. So we use zebrafish for these studies, and I hope from Erica's talk you realize this is not an actual zebrafish here. Our strategy has been to generate first loss of function, strong loss of function mutations, and take advantage of the things that we can do in zebrafish because of the external development, the optical clarity, very strong genetics, to study the underlying biology. And one thing that I didn't list here that I think is really critical for zebrafish as a model is that it's a really excellent system for high throughput drug screens. And that's because they're very permeable, or the larvae that we use are very permeable to small molecules, so we can just drop drugs into the water and they take them up. And so that really increases the throughput of what we can do. And lastly, we're trying to develop humanized zebrafish models, so to knock in disease-associated variants to complement the in vivo studies and determine pathogenic mechanisms. And these actually would be really excellent substrates for small molecule screens. Okay, so one of the first questions is, well, in order for zebrafish to be a good model, we really have to know that the NMDA receptors function very similarly to mammalian NMDA receptors. And so, as Erica already mentioned, the sequence similarity is very high. We've also looked at functional similarity. This is through expressing NMDA receptors in HEC293 cells. So this is actually rat, mammalian, GLUN1 and 2A, given a pulse of glutamate. And you see this very nice induction of current and decay kinetics. And now these are two zebrafish paralogs for GRIN1. We see that the physiological properties of these, and among others, are virtually indistinguishable between the zebrafish and the mammalian receptors. Okay, so we've made mutations in all of the principal NMDA receptors in zebrafish using the CRISPR system. As Erica alluded to already, zebrafish actually have 2 copies of each of the subunits, and they're designated by these A and B paralogs. And there's some subtle differences in the phenotypes that we see between the mouse mutants and the zebrafish mutants, although the phenotypes are overall similar. So the 2 key differences, in the N1, for the whole knockout, the mouse mutant is perinatal lethal. In zebrafish, these animals survived about 10 days post-fertilization, which gives us an opportunity to both look at later stages of neural development. Also, there's a lot of interesting behaviors that we can look at during this time window. The other difference, big difference, is that the 2B mouse knockout is a perinatal lethal due to a suckling defect. Zebrafish don't have to suckle, and that gives us a little bit of an advantage. And so the 2B mutants, probably for that reason, are partially viable, and some of them will survive to adulthood. We began by looking at I'm going to talk to you today mostly about our N1 and the N2B efforts. We began by looking at the brains of the GLU-N1 mutants. And this was work done by a former PhD student, Amalia Napoli, in my lab. And what we noticed very early on is that there were very obvious differences in the number of neurons in GRIN1 mutants compared to wild type. So what you're looking at here, this is the sagittal section across the telencephalon of a 5-day zebrafish larva. So these are the eyes here. This is the brain here. So this is the wild type. This is a mutant, and you can very readily see that in the mutant there's more cells there. Right? This is a Nissl stain. It actually marks both neurons and glia, but we know that through subsequent marker analysis that the ectopic cells are neurons, or at least many of them are. This is just a comparison at 9 different levels across the telencephalon, comparing wild type in this dark purple to our GRIN1 loss of function animals, and just looking at the cell density, and we see that in all regions in these GRIN1 mutants, we see considerably more neurons. The reason that we think there's more neurons is because there's an excess of proliferation. So here's looking at a little bit earlier, at 3 days post-fertilization, at a marker for proliferation, PCNA, comparing a wild type, again with our GRIN1 double mutants. And again, this is a sagittal section through either a more rostral or more caudal region of the telencephalon. But I think it's pretty obvious. You see that there's a lot more PCNA positive cells in the double mutants than in wild type. And if we look at more carefully at where we're seeing these overgrowths of neurons, what we noticed was that the radioglial cells are these cells here that are lining of the ventricle. And we don't see a lot of differences in these in radioglial cells or neural stem cells. However, the cells that are a little bit farther away are what's referred to as transient amplifying cells or neuroblasts. And it's really in those cells that it appears the defects occurs. And through additional analysis that I won't show you today, really think is happening is that these transient amplifying cells or neuroblasts are failing to mature, and they're remaining in a proliferative state for a longer period of time. So as many of you probably are aware, that autism is associated with an overgrowth of prefrontal neurons, so we're very excited by this finding. And we wanted to ask whether there were behavioral defects in the fish that might be in some ways reminiscent of autism. Excuse me. Now, the GRIN1 mutants only survive till about 10 days, which isn't really so we're not able to look at social interactions in those animals. However, as I mentioned to you, the 2B mutants do survive longer, and it allowed us to look at social behaviors. And in fact, we've identified social deficits in the 2B mutants. So very briefly, the assay that we do utilizes this kind of arena where there's 10, it's a high throughput assay. So we're able to there's 10 lanes where we're able to assay one fish per lane. So these fish can't actually see the neighboring fish here between the lanes. What they can see is at the end of each lane, and at both ends, indicated by these dotted lines, is a window. And we put either a conspecific wild type fish or no fish behind the windows, and then simply ask, much as you would do in a rodent assay, if this fish has a social preference. Does it prefer to swim closer to a conspecific fish? And arbitrarily, we've just divided this arena into 4 zones. So zone 4 is the social zone near the conspecific, and zone 1 would be the farthest away. So if we look at wild type fish, and this is over a period of 4 weeks, so this is weeks post-fertilization, what we see is that somewhere between 2 and 3 weeks, as it's been observed by others, the fish start to develop a social preference such that they now prefer swimming near a conspecific. And that increases a little bit further at 4 weeks. And so this is the same data just shown in a slightly different way. We've converted this to a social preference index, which is just a measure of distance from the conspecific. And we see here that wild type developed this very nice social preference. But if we look at our GRIN2B mutants, what we see is that they have a severe social deficit, reminiscent of what might be observed in autism. We don't think that these are autistic fish. Obviously, human behavior is much more complex, but this does suggest that some of the circuitry underlying social behaviors in both zebrafish and humans require GRIN2B. One of the features associated with autism, also with epilepsy, is an imbalance of inhibitory and excitatory signaling. So we wanted to ask, and sort of really take advantage of the kinds of things we can do in zebrafish, to ask whether these 2B mutants have an imbalance in inhibitory and excitatory neurons. So what you're looking at here is a series of confocal sections going from dorsal through ventral through an animal that's a double transgenic. In red is V-glute, so these are excitatory neurons. In green is a DLX marker that's labeling inhibitory neurons. This is the optic tectum here. The telencephalon is positioned a little bit more ventrally, so it's only going to come into focus into some of these deeper sections here. And you'll start to see now some of the interneurons of the subpallium and the deep layers within the telencephalon that come into focus. So we've done this with both wild type and mutant animals, and what we see is that, let me just stop that. The excitatory neuron populations don't appear to be altered, but what we do see is the inhibitory neurons, particularly in the subpallium, are reduced in the GRIN2B mutants. And it turns out when we dig deeper there, we see is that the regions where they see these deficits correspond to things like the amygdala, striatum, and septum in humans, which are also regions where you see interneuron deficits in humans with autism. So we think there's both construct and face validity to this model. As has been talked about elsewhere today, variants in 2B and other subunits have diverse functional and phenotypic consequences, and it's not always clear why. So for example, here you have 3 mutations in the M4 domain that are very similar and very close to each other, and the effects on function are different, and the phenotypes differ slightly too. And this suggests that while obviously the heterologous expression in HEX cells is very valuable and we can learn a lot, there maybe are some things that we need to learn from whole animal models of NMDA receptor variants. And so we've been setting about to develop a large collection of zebrafish lines that harbor patient-specific variants. And we're using an approach called prime editing. That's a new technique for genome engineering. It's a CRISPR-related approach that, very briefly, and I'm happy to talk about this in more detail afterwards, contains a Cas9 nickase that's fused to reverse transcriptase, and you utilize a special guide RNA called the PEG RNA, a prime editing guide RNA that contains these 3' sequences that contain, shown here in green, this primer binding site that binds to the Nick strand and serves as a primer for reverse transcriptase that then allows reverse transcriptase to add sequences to the Nick strand that can be incorporated into the host genome. So what we've been doing is we've been injecting these prime editing complexes into single-celled zebrafish embryos, and we can assess somatic mutation frequency and screen for germline transmission. This approach, so we've been targeting in our initial effort 5 different mutations, one in GRIN1, this M706V, GRIN2A, L812M, and then 3 different mutations in GRIN2B. As a first step, this is a very new protocol. So we wanted to optimize what works well in zebrafish. We tested a number of different prime editing proteins, and it turns out that this PEMAX one in particular, so this is looking at the rate of pure prime edits, so it's the efficiency of it at these 5 different loci that are color-coded, and this PEMAX works very well, and we've obtained a high degree of targeting at 4 of the 5 loci, where we've also been able to obtain germline transmission. This is just showing the data for one of those. So the question is, and so we're very much interested in looking at the phenotypes of these animals. And so for this mutation, this GRIN1 M706V, we started looking at social behaviors. In this case, the two paralogs create a little bit of a complication for us because we essentially have 4 copies of each of the subunits. So in this case, we're looking at animals that are homozygous for M706V in the GRIN1A allele, but wild type in the B. We don't see a social deficit. We see a slight effect in animals that are heterozygous for M706V, but now lack both copies of the B allele. However, if we eliminate the GRIN1B paralogs and we make both of the GRIN1A paralogs mutant for M706V, we now see a very strong social deficit. So we're very much interested in diving into these further, looking at how neurogenesis is affected, looking at if they're susceptible to seizures and such things. So in conclusion, we see deficits in GRIN1 that include supernumerary telencephalic neurons due to a failure of maturation of neuroblasts. Our 2B fish display social deficits and deficits in interneurons, similar to what's seen in autism patients. So we have both construct and face validity, and we've begun to assemble a large collection of fish lines harboring human NMDA receptor variants to be able to stratify variants. And we already see that some interesting phenotypes in M706V. So I'd just like to thank the people who actually did the work here. So the gene targeting has been done and the variant development by PhD student Gina Rizzo, along with a technician, Rahman Basharat. The 2B model was developed by a former PhD student, Josiah Zutzma. The GLUN1 neurogenesis analysis was done by a former PhD student, Amalia Napoli, who's actually now a postdoc in our close collaborator, Lonnie Wolmuth's lab. Yeah, I'll just stop there. Thank you. Good questions.
Excellent. We're going to take a quick break from the zebrafish, but we'll be right back to it. So hang in there. So next we're going to invite Dr. Calliope Hollingue up to talk about an overview of gastrointestinal symptoms among individuals with neurodevelopmental conditions. Welcome.
All right, good afternoon, everyone. It's a pleasure to be here, and thank you to the CureGRIN Foundation for the opportunity to speak. So, as you just heard, I'm going to mix it up a little bit. This is going to be focused on humans. We're going to take a much sort of broader view of things. All right. So I think you all know that this is going to be not so much focused on GRI conditions specifically, but neurodevelopmental conditions more broadly. A lot of what we see in GRI conditions is very similar to what we see in a lot of other NDDs. So I think this talk is still relevant. But as you all know, the health of individuals with NDD is very complex. NDD individuals often have complex health needs. This population tends to have greater use of healthcare services, more emergency department visits, more inpatient hospitalizations, higher costs. And despite this greater use of health services, this population broadly has a lot more unmet health needs, greater issues with accessing quality health care, lower quality of care on average. And I really see this as an equity issue because, as I'm sure you can all understand, when someone isn't feeling great physically or mentally, that is going to affect all domains of their life. And so even in someone with a GRI condition who may have other sort of impairments, having untreated, unmanaged GI symptoms can really pose a greater sort of burden on the individual and their family. And this schematic here on the right is specific to autism, but I think the point is relevant more broadly, is we have an incredible amount of heterogeneity in the NDD population, in the GRI populations, in terms of both mental and physical health needs. And that's important to consider in terms of GI symptoms as well, because you'll have families in which the GI system doesn't really seem to be too involved, and others in which it might even be the main problem that the family is encountering. All right, I'm having trouble advancing to the next slide. There we go. Thank you. So we do know that GI symptoms are incredibly common in neurodevelopmental conditions. This is from a review paper that we wrote a few years back at this point, but we looked at studies between the years 1980 and 2017 in autistic individuals, and we saw that nearly half of individuals across all of these many dozens of studies had at least one GI symptom. And when we look at sort of the NDD population more broadly, we see that individuals are over 4 times more likely to have GI symptoms compared to neurotypical individuals. And this is the trend that we see in GRI conditions as well. Some of the most common symptoms that we tend to observe are constipation, diarrhea, abdominal pain, reflux, and encopresis, essentially stool leakage around a stool impaction. And as you can see here on the right, this is just a plot showing that across different studies, there is variability, as you might expect, in terms of the prevalence of these GI symptoms in the various populations. But on average, we do see this pretty consistent elevation. Next slide, please. All right. And as you might intuit, there are a lot of correlates of GI symptoms and GI pathology, which can make things even more difficult. So food intolerances and toileting difficulties are incredibly common. When someone is having something like abdominal pain or pain from constipation, that can lead to sleep challenges, a lot of behavioral dysregulation, things like anxiety, irritability, even aggressive behaviors. We even see some from research and more anecdotal stories that seizures can occur or can be exacerbated due to untreated constipation. And often this can lead to greater emergency department use, and a lot of really traumatic healthcare experiences in the pursuit of trying to treat these symptoms. Next slide, please. All right, and then more broadly in terms of impact of these GI symptoms on the individuals with the NDD themselves, this can lead to poorer quality of life. So a lot of unpredictability, a lot of pain, difficulties with social outings or other activities. It can really influence the diet because people are so desperate to try to figure out what can I eat or what can my child eat to try to resolve this symptom, and that alone can sort of take on a life of its own. And then for individuals who are able to be employed, GI symptoms can lead to challenges with finding and maintaining a job, for staying in school, etc. And then more broadly on the family, this has impacts on the overall well-being of the family, the household. Parents' mental health we see is very much tied to how their child's GI symptoms are doing. The ability of the family to go out, even the family's finances, because families end up spending a lot of money trying to manage these symptoms and try various things. So we see that financial stress is not uncommon as a result of specifically the GI symptoms. Next slide, please. All right. And this, I think quotes are so powerful, and qualitative research is such a helpful tool. This is a quote from a parent of an autistic adult, and it reads, "When the stomach hurts, there's an almost immediate physical reaction. She could scream, she could throw things, and after she calms down, then she tells us, 'My tummy hurts.' Every time the police had to come out because she was being really destructive, first thing she would say as she regained her composure was, 'My tummy hurts.'" So I hope this quote convinces you of not only the profound impact that these GI symptoms can have on the individual and the family, but also this underlying issue of if the child is nonverbal, or has limited verbal ability, it's going to be really hard for the family to understand that these GI symptoms are going on. Sometimes it's obvious, but a lot of times it's not. And so that just adds this whole complicating layer. Next slide, please. All right, and this quote is from an autistic adult that we interviewed about their experience with GI issues. And he said, "I don't know if I'll be able to hold down a full-time job or not. Will I ever be able to date or do anything?" And just to clarify, he's referring specifically to concern because of his GI symptoms, not his autism or anything else, but his GI symptoms were so severe that he didn't know that he'd ever be able to hold up a job or date for that reason. So for some people, these are truly disabling. Next slide, please. As I'm sure many of you are familiar with, particularly if there are families here listening, GI healthcare and healthcare more broadly can be a compounding issue to all of these challenges. So there's often a shortage of trained and confident providers. And I say confident because someone can have sort of the best training, but if they haven't really had exposure to individuals with NDDs, they're not going to feel confident treating that population, and they might be inclined to say, you know, I'm just not the right person for you, and I sort of don't want to get involved. And that's something that we hear sometimes from clinicians. The approach to treating GI issues and NDDs are not always clear, so it's a complicated field and we don't always know the right answer. There can be communication challenges between the patient and the clinician, and then communication challenges more broadly between the family and the clinician. Diagnostic overshadowing is unfortunately quite common, in which a person is sort of told, well, this just goes with the condition that you have, or this is due to perhaps anxiety, and there's really not much we can do because it's really not its own thing, it's due to something else. And that's something we hear quite a bit in the GI world as well. And healthcare settings are often not conducive to individuals with NDDs. So things like the sensory environment may be very overwhelming. And so getting someone to that appointment is already a whole challenge in and of itself. And speaking to the point of diagnostic overshadowing, this parent of an autistic child shared with us, a lot of physicians seem uninterested. It's sort of like, your child has autism, the GI symptoms, go with it. Next slide, please. All right, so one study we did recently is we worked with Spark. This was funded through the Autism Science Foundation and the Autism Intervention Research Network on Physical Health. And we carried out a mixed method study in which we spoke to both autistic adults as well as parents of autistic adults about their child's experiences with GI symptoms. And we asked for their recommendations on what would be the most helpful things in order for the field to move forward. And I recognize some of these may not be as applicable to GRIE conditions, but I think a lot of them are. The top 5 emerged as 1, a need for healthcare provider trainings on how to interact with this population. 2, a healthcare advocacy training. In this case, it was advocacy training for the autistic adult themselves, but I think probably more relevant to this population would be healthcare advocacy training for the family in terms of how to navigate the healthcare system and be better advocates for the child. Make sure that they're getting everything they need. An interoception intervention came up as something that would be helpful. So recognizing that a lot of individuals with NDDs have difficulty sort of naming what they're feeling and communicating that. Interventions focused on saying, okay, this is what GI symptoms feel like for your child, and this is how to communicate it. That would also be very valuable. Because we know that anxiety is often a trigger for GI symptoms, anxiety interventions were recommended as sort of a next step. And then number 5 is very broad, but care coordination. So some kind of interdisciplinary model in which all of the providers are talking to each other, care is in one place, and the family is more able to navigate the healthcare setting. All right, so there are a lot of challenges with GI assessment, which I've alluded to. The studies that we've looked at show that there's a very wide range of GI symptom estimates across populations. This is due to not only phenotypic heterogeneity, but also due to measurement challenges. And so, for example, the autism review study that I spoke on briefly at the beginning of this talk, the estimate for diarrhea ranged from 2% of the population to 75% of the population. So you might look at that and think, well, that's practically meaningless. What does that possibly tell me? But what we found is that because of all the different ways that symptoms were measured, and because of the limitations of how these symptoms were measured, that actually influenced the symptom estimate itself. Oftentimes, the population questionnaires that are used in autism studies or studies of individuals with NDDs more broadly are not really valid. And that's because of these impairments in interoception, because of communication and language difficulties, and because of intellectual disabilities. So the tools that we sort of take for granted and in the general population just often don't work here. So that has led us to a number of collaborations in which we're focused on improving assessment of GI symptoms in NDDs. This is not my work, but a work of a close collaborator, Kara Margolis at NYU. She's a pediatric GI doc who focuses on the NDD population, and I'm just highlighting her paper here of a brief parent report screen for common GI disorders in autism. Oops. And this is a questionnaire that I have led in which we're also focused on GI symptoms in autistic children. And we now have an NICHD R21 in which we're focusing on broadening this questionnaire across the life course and making both self-report and parent report versions. And ultimately, we want to create versions that it can be used across the whole NDD population. I also want to highlight some work that's been funded by the Phelan-McDermott Syndrome Foundation and by CureGRIN. And these are our team members here. Julia Dallman is here in the audience, and she'll be giving the next talk on her zebrafish work. But basically, with the recognition that GI symptoms and GI physiology is difficult to assess in this population, we have and mainly Julia Dallman and Baha Moshri have worked to develop and refine a recipe for a blue muffin in which there's blue dye food coloring in the muffin. The child with the GRIE condition or with PMS can consume that muffin, and the parent can track the stool coloring, and we can use that as an indicator of GI motility. In parallel with this, we have been developing an app called the Stripes app. This is really spearheaded by Dylan Baker and Dr. Barbara Millay. And this Stripes app has been refined for individuals with NDD. And that's one of the focuses of the grant that we have from the CureGRIN Foundation. And then just to close, this isn't the focus of my talk today, but I do want to just briefly speak to what are the potential mechanisms here. We know that ASD and NDDs more broadly increase the likelihood of having GI symptoms. Why might that be? So one sort of obvious answer is particularly in individuals that have more of this autism phenotype in which there's a lot of sensory factors at play. The diet is often driving some of these issues like constipation. So if you have a child that is not eating anything with fiber, that is going to cause or exacerbate certain GI symptoms. Similarly, we know that co-occurring conditions or impairments in terms of mobility are going to cause or exacerbate GI symptoms. So someone that doesn't have the ability to move very well isn't going to have that same motility as someone who is able to move around. In the focus group that I conducted right before this talk, the idea of like the standing frame came up as a way of helping some of this constipation in individuals that aren't usually able to move around. And then medications, of course, can be, side effects due to medications, which are taken, for a number of reasons, can also contribute to these GI issues. And then beyond that, we have sort of more of the biology. So things like gut-brain axis dysfunction, dysmotility, alterations in the immunologic system, gut microbiome, and overall this points to a need for more etiologic research. So with that, I just want to thank the participants of this work, the collaborators, and importantly, the funders. And I'd like to particularly thank CureGRIN Foundation for funding our study. And we're very excited to move this forward and to work with families to find some solutions. So thank you so much for your attention.
Okay, thanks so much. We're going to finish up this section and do a Q&A right afterwards. So now we're going to introduce Dr. Julia Dallman, who's be talking about assessing the role of GRIN-2B in gut physiology using zebrafish models. Thank you.
Okay, thanks so much. Ta-da! Okay. Thank you so much for your continued attention. Thank you to CureGRIN for inviting me to give this presentation and for funding this project that we're very excited about. Disclaimer before we start, we're getting into it. So I'm going to talk about work in other models as an example of what we plan to do with GRIN-2B. Okay, so as has been mentioned, GI distress and molecularly defined ASDs and NDDs are common and have negative impacts on quality of life, as Dr. Hollingue so beautifully described in her talk. This is a list of some of the conditions that have, and it's just a subset, but some of the genes that are associated with significant GI distress. At lunch, I learned that CAMK2 should be on here as well. And the list continues to grow. As Dr. Hollingue said, because those symptoms are difficult to assess in their interoceptive symptoms, these are not trivial to really understand in these populations. So common GI symptoms, so this is kind of repeating what just came, but feeding problems, constipation, reflux, cyclical vomiting. Gastrostomy tubes are very common to address. So as Amy Ramsey said, those GRIN-1 mice were not growing quick or were underweight, and the gastrostomy tubes can be indicated in cases where there's failure to thrive. And then there are also links between GI symptoms and either self-aggressive or other aggressive behaviors, sleep, and seizure. So a big question, is this dysregulation of the gut by altered CNS? Because gets lots of innervation from the CNS through the vagus and the spinal neurons, or are there gut intrinsic mechanisms? And so my lab has decided, has sort of chosen to focus on the gut intrinsic just as kind of a peripheral area that we thought was kind of understudied. So first I'm going to take you through some of so here, this is a paper that did single cell sequencing from colonic tissue, the enteric neurons from colonic tissue, and on the sort of the x-axis are a list of genes that have been linked that are neurodevelopmental genes, and you can see that they're expressed in this peripheral tissue. Likewise, this is a study in mouse that again has a whole list of genes and showing that they're expressed in the digestive tract. GRIN-2B is not listed on in these papers. I guess I have a question for the audience. So these are channels, receptors, and single cell sequencing can be a little shallow in terms of its depth of So I think that an absence of GRIN2B in these peripheral tissues, I'm not sure how seriously to take it. But so we have to take these with a grain of salt. But clearly from Dr. Ramsey's talk, pancreas is a site that is a peripheral site that has expression of the glutamate receptors. Okay, so one of the reasons that it's really nice to study GI physiology in zebrafish models is, as Dr. Sirotkin so nicely introduced, we really think of these larval stages as a sweet spot because these fish have precocious development. So by 6 days of age, they can feed on their own, they can escape predators, and you know, so they take part in a lot of the physiological functions, and yet they're transparent. So this is a blow up of a zebrafish on its side, and it's been fed fluorescent beads, and you can easily see the motility of the gut as those beads are tractoring through the intestine. So this accessibility makes GI physiology really nice to look at. Okay, so I did not make the GRIN2B mutant. As you heard, Dr. Sirotkin has been making this beautiful array of glutamate receptor mutants in zebrafish, and we now have the GRIN2B model at UM, and it's an adult, so we can start crossing it to all of our physiological lines. In the meantime, I'm going to tell you about what we've done with some other mutants so that you can kind of get a taste of the sorts of experiments we do. Okay, so I showed you that movie of the beads moving through the gut. These are 3 little movies of fish that were recently fed. So in the wild type, you can see propagating this really nice example of peristalsis, propagating waves of contraction going down the gut that are lacking in 2 of our models of conditions that are linked to NDDs. So Shank3 hets are our model of Phelan-McDermid syndrome, and SYNGAP is our model of SYNGAP1-related disability. And the smooth propagating waves are gone. We can quantify those peristalses so on the y-axis is time, and on the x-axis is space. So what you're seeing is these propagating contractions moving down the intestine. So yellow pixels mean that there's more movement, and so these movements come in waves that have very regular intervals. So we can measure those intervals, and you can see Shank3 has a longer interval between contractions, whereas SYNGAP is spot on to wild type. And so even the way that the GI physiology is affected is distinct in these 2 models, and I suspect we'll find similar distinctions in other models. This is not something that's only been found in fish. There are many mouse models that have also shown GI dysfunction linked to neurodevelopmental conditions in Shank3, FOXP1, neuregulin 3, TCF4, CHD8, and others. And so one of the ways of doing a more careful quantification is by feeding these beads, and what that shows is that there's accumulation in the intestinal bulb, which is the equivalent of sort of our largest chamber of our GI tract, which is our stomach. So there's delayed stomach emptying in these zebrafish models. And so we plan to do similar experiments in the GRIN2B model to see if they have reduced motility. There are pro-motility drug therapies that could potentially alleviate symptoms. And as Dr. Sirotkin said, the fish are a really nice model in which to add small molecules to see if to test those compounds. So that says that there's a motility problem. What are the cells that mediate motility in the gut? Well, there are these gut intrinsics. There's this gut intrinsic circuit. So your gut is lined by smooth muscle cells that are innervated by, the gut has its own nervous system, the enteric nervous system, that comes from those wonderful neural crest cells. And those are talking to these sensory endocrine cells, which are in light blue, which are cousins of the pancreatic cells that are secreting insulin and glucagon. They actually derive from the same tissue. It's a bud off. And these guys are like, they're your largest endocrine gland in your body, but they're scattered across the epithelium. And they're the only way, they're sort of the reporters of what's in the lumen of the gut. So in trying to understand, so I should say that things like bulk can activate subsets of these enteroendocrine cells to induce motility. So like bulk induces motility and push things along. But there are also sets of those endocrine cells that are sensing nutrients and put stomach emptying on pause until those nutrients have been absorbed so that you don't get rid of the nutrients you did so much work to collect, like we just did at lunch. So, in looking at these cell types, SYNGAP shows a sort of loss of enteric neurons, especially in the posterior end of the intestine, whereas the Shank3 model shows a dose-dependent loss of these enteroendocrine cells, or those sensory endocrine cells. And in particular, the sensory endocrine cells that are responding to bulk and bacterial pathogens to increase motility. So those are the enterochromaffin cells. And so we also wanted to better understand the physiology of how those cells are sensing. And so this is a tool that was developed by John Rawls' lab at Duke, actually Liu Huaye when she was in his lab. And the NeuroD1 is used to drive a genetically encoded calcium indicator that stably converts when calcium goes up and you shine a UV light on it. So what we do, all of the enteroendocrine cells have the Campari. We feed them glucose. We shine UV light on 5 minutes later. And then here in wild type, you can see the cells that are unconverted are green. The cells that are converted also have some red. And so you can see the cells that have responded to the glucose. And in our models, we see a failure to respond to glucose. So glucose is a theme that keeps coming up. This is an actual movie of them responding to glucose in real time. So here's the pancreas in wild type and the enteroendocrine cells that are sprinkled across the gut epithelium. And when they turn cyan or red, that's calcium transients going on. This movie in SYNGAP is also playing. So the enteroendocrine cells are there. They can produce signals, but they're not responding to the glucose in the same way. And this was work that was spearheaded by Elizabeth Davidson when she was in my lab. This was a surprise for us. We were kind of being formal circuit busters. We wanted to look at the sensory component, and so we wanted to look at other stimulants of these enteroendocrine cells. So another thing the enteroendocrine cells respond strongly to is the bacterial metabolite indole. And these SYNGAP guys have a beautiful response to indole across the same concentration range that the wild type 2. And so we plan to do these similar experiments in GRIN2B for the sensory, what the enteroendocrine cells are sensitive to. And if there's altered nutrient sensing, this might be addressed with something like diet. So there are some interesting convergences. Okay, so I want to acknowledge the people who contributed to the work. So it's really wonderful that Howie was so generous to send us the model. David James was the first person to get me into the gut. So, and then Elizabeth followed closely in his shoes. John Rawls is at Duke and has really also sort of supported us going into the gut, and Li Huaye as well. Mason Klein helped with the peristaltic analysis, and Rob Kozel made all the models that we've studied. And then this is the same group of pictures that you saw in Calliope's study. But with GRIN2B, we're doing this integrated analysis where we're trying to better understand the GI symptoms in people, and then also look at the underlying physiology in the fish models in GRIN2B. So thank you so much for your attention.
If our speakers want to join us up there.
And I give them this one and I hold on to the, oh, it doesn't matter.
All right.
Hi there. I've got a question for Howard about sort of one or two of the last slides that you were showing with the variant that you created, and I was just wondering, you created them in the A paralog, I think.
Yeah.
And I was just wondering, what's the relative expression like of the A and B paralog? Is the promoter stronger or weaker in one than the other? And have you tried doing it the other way around?
Yes. So that's a good question. And ultimately, for some of these, we'll probably do both paralogs, because the paralogs do really complicate, especially the variant analysis, right? Because the humans are hets, and now we have 1/4. It's actually nice because we can make an allelic series. We chose the 1A paralog for GRIN1 because it's expressed a little bit earlier and a little more robustly than 1B is. And in terms of the behavioral phenotypes, we tend to see stronger phenotypes in 1A than in 1B in most cases. Actually, interestingly, the 1A strong loss of function mutants, they survive to adulthood, but they're actually very small and runty, which we thought might be a feeding defect, but it could also be obviously related to pancreas. Curiously, they recover over time. As they get to adulthood, they catch up. But yeah, we chose the A paralog because it's expressed a little more robustly and a little bit earlier.
Yes, thank you so much. I have a question about the gut motility indeed in the zebrafish. And the expression patterns. I'm very happy indeed that you mentioned that CureGRIN should be on there, because I do think that CureGRIN should be on there. And you show there a full list of genes, but you mentioned that the GRIN2B was not expressed in the anterior nervous system. How about SYNGAP and SHANK3? Do you know whether they are expressed in the...
So it depends on the model. And that's why I'm saying I think there's some false negatives in the different data sets. Because, in zebrafish, SHANK3 is not in the ENS, but it is in the enteroendocrine cells, and SYNGAP is the reverse.
Okay.
But in mouse, both have been shown to be expressed in the epithelium. So there are some inconsistencies across models, and it's not clear if it's how they looked or...
No, because what I find very interesting is that they are all substrate for CureGRIN, and we've done stainings looking where CureGRIN is expressed in zebrafish, and we do find it also in the anterior nervous system. And if you indeed overexpress there at hyperactive, you get clear motility issues also in zebrafish. So I think this is very, very fascinating, and I wouldn't give up on a GRIN2B.
No plan to give up on GRIN2B. No plan.
Okay,
so
I really have to.
So what we're doing, so single cell seq tends to be a little shallow and have false negatives. We're doing a deep sequencing, and so we'll know.
Good.
We'll know soon.
We will be very interested in knowing this.
Yeah.
Thank you.
Yes.
Sorry, I'm the clinical colleague for the ChemK2. I was just wondering how well can in zebrafish can you also model the difference, like the different GI tract compartments, right? When I think of the children, like sometimes they have like subgroups that really have upper GI dysmotility, that stomach release emptying problems, and then the other ones are completely more constipated. And sometimes it's within a group, and I was wondering, like, can you elaborate a little bit how maybe the zebrafish is also a good model to figure that out?
Okay, so there are some places where zebrafish is, well, so one thing I would say is that this is a sort of beautiful convergence of where the basic science is making big headway, just in terms of like vagal regulation, spinal neuron regulation of the GI tract. All those things are actually emerging fields, I would say. In terms of the correspondence between the zebrafish regions and the mammalian regions, there's some we can't call that intestinal bulba stomach because it's not an acidified compartment. But in terms of motility, it behaves very similarly. And indeed, if you look at those bead movies, it even looks like they're segmenting their beads into poops, you know. So likewise, the colon, it's not drying out the poop because fish are freshwater animals and they have to rid themselves of water all the time. So there are some things that are completely different, but I think they have very similar sets of hormones. They have very similar sets of feedback. There are cholecystokinin cells that trigger release of enzymes from the pancreas. So there are a lot of parallels, and I think those we can make good use of.
Yeah, and a high throughput scheme for better medication, right? Because we have so few to do, things to give to the patients also, right?
Yeah.
Besides the laxantia and all that. So yeah.
Thank you. Those were all great talks, and appreciate the science you're doing. I'm curious, Julia, how does glucose sensing work? Like, is it a glucose what binds glucose? Is it a transporter?
So it's a sodium glucose co-transporter.
Okay.
That's thought to depolarize those cells.
Okay.
And so that's another reason we're doing the deep sequencing in the mutants versus
right.
is
to find that.
To find the molecules that.
Okay, but what is the gene? What is the gene name? Do you know, or SLC?
I think it's SGLT1.
Okay, thank you. I want to look and,
Yeah.
I just want to see what's going on.
So there's SGLT1, and then there's also some GPCRs.
Okay.
That
sense glucose. Wow.
Yeah.
Cool.
I have a quick question for Howie. So totally understand why you're unable to do social behavior in these mutants here. But I'm wondering if you can do, or that specific social assay, but could you do something like schooling or shoaling, or are they still not useful?
So for the N1, GRIN-1, because they died at an early age, right around the time when social deficits occur. So we're actually, rather than doing a purely gene knockout, we're developing a GRIN-1 line and a 2B line that have an epitope tag that allow us to do protein-mediated degradation, an antibody-mediated protein degradation, so like a conditional knockout. So for those we could. For all the other ones, we've done it for each of the single GRIN-1A. It's so confusing, the nomenclature, because in zebrafish we have GRIN-1A and GRIN-1B, and then we have GRIN-2AA and GRIN-2AB, and so we're just always confused about these things. But we've looked at the GRIN-1A and GRIN-1A single mutants. We don't see a social deficit. We don't see a deficit in 2D either, or 2A. It's really specific to 2B, and presumably we would see it potentially with N1 also.
Great.
Yeah. But there are actually a lot of other behavioral assays that I didn't talk about in terms of learning and stimulus responses and stuff that we can do on those fish.
Great.
This is a question for Calliope. So it seems so complex trying to measure motility in humans, and then you add on the special needs part of it, and then you add on the lack of verbal communication, or sometimes even gestural communication that our kids have. Okay, so you add that on, and then you're trying to measure motility that's kind of caused, changed by everything, bacteria, what they eat, even psychological withholding and pain and so on and so on. And then the layer of medications, right? A lot of the anticonvulsants and the mood stabilizers, they really affect, motility. So anyway, my question is, how do you approach it given all this, like this sort of, this complex network of things that affect it?
Yeah, good question. So one thing I didn't mention is that in this CureGRIN grant that we're doing, there are a few pieces, and one of the pieces is essentially a long survey asking about all sorts of clinical information, including medications, some GI stuff, some dietary stuff, and we're trying to use that to see if we can find correlates of GI motility, not only for sort of validating, but also for understanding what are things that drive it. So this is sort of a little bit of a pilot grant. We're not going to get all the answers, but I think ultimately in combination with the Stripes app, what we'd love to do is a little bit of like ecological momentary assessment in which people are tracking lots of information through the app as well as the motility information, and we can start to tease out, okay, what does it look like across the GRI conditions? How is it influenced by various things? We're still a little bit a ways from that, but I think we're setting the foundation for that future work.
I really hate to cut off a fruitful conversation, but we are behind on time already. You guys will be around, presumably. Feel free to ask them questions later. Thank you guys so much. Don't forget your books. Here, a book for you.
Thank you so much.
It's a book.